when it comes to this formation of a sperm or an egg, we are assuming that everything happens normally. Okay. And for all intents and purposes, most times it does. But there are a number of times where it can screw up. And the older you are, the more prone you are for these improper mitotic divisions. Okay. We have some terminologies that, again, would make a good maybe matching question or multiple choice question on an exam. Euploidy and aneuploidy. This is the correct number, and this is an abnormal number. Okay. Euploidy is normal number of chromosomes. Or euploidy is normal number of chromosomes. Aneuploidy is an abnormal number of chromosomes. That can be one too many. That could mean one too few. Okay. It is just a not proper number. Okay. Not proper number. And I have some slides here that will kind of show that off. I kind of step it out step by step there. Aneuploidies result from this failure of the chromosomes to separate, and we use this term called non-disjunction. They didn't separate. That's all that says. Okay. Non-disjunction can occur really during any time, meiosis 1 or meiosis 2. And they're both potentially bad. Now, not every aneuploidy is bad, okay, or resulting in a non-viable offspring. Okay. There are a number of them that grow up. The most common one is trisomy 21, right? Down syndrome. Both viable offspring, male and female, but from a reproductive capability, the males are not, and the females are. Okay, with that aneuploidy. To get a visual representation of this, we can see what the chromosomes look like, and that is called a karyotype. Okay, and there are different ways to check the genetic material. All of them result in a slight chance of spontaneous abortions. So all of them have an inherent risk. And if this is something that you want to check out, there is some inherent risk to kind of understand because it can destroy the capability of maintaining that pregnancy. So here's what some of these non-disjunctions look like. Okay, so here we can see we have a normal kind of homologous. Well, here we can see in meiosis 1, we have one side where it was normal, and then we can see the other one where those chromosomes did not separate as they should have. One should have gone to each pole. For some reason, it didn't do that. You can see both went to one pole. So I have one of these with one too many, and I have one of these with not a dang thing in it. So obviously, if that one on the right, this guy right here, is the one that happens to, become the mature egg, okay, that's not a good situation, right? So I have two with absolutely nothing in it. Okay. Here you can see non-disjunction occurring in meiosis 2. Obviously, we only have one potential bad egg here. Okay, or bad sperm, whatever you want to use. All right. Obviously, if this and this were the ones, these two in this position here, obviously it wouldn't work. Okay, wouldn't work. There's no genetic material there. All right. But if this one merged with that one, then there's your monosomy. Or if this guy right here merged with that guy right there, there's your trisomy. Okay, has extra. And that's what they're trying to show you off here. Okay. If those eggs did get fertilized by a sperm, you can kind of see what it would be. Okay. Normal, obviously normal. Trisomy. 3, right? Monosomy. Monosomies. Trisomies. Again, one of the most common ones, guys, is trisomy 21. It has a number of inherent characteristics: short stature. They have a kind of a simian grip, wide kind of gap between the first and second toes, like a simian. They have kind of a much kind of broader, slightly stubbier hands, look more simian in nature, flatter face, different eye folds. There's just a number of things that are inherently recognizable with this thing, with this condition. Again, more common in women producing Down syndrome children just because eggs are older. So there's going to be a lot more issues with them having issues with non-disjunction. That's why understanding your cycle, when you start, when you stop, is important to kind of understand, because it's from those dates that they determine how far along you are in your pregnancy. And when you, at certain points, there are certain markers that are going to be elevated, certain protein secretions that are going to be, elevated. And if you don't know your cycle, and you don't have because Jennifer, she didn't have a regular cycle. Sometimes she was 4, sometimes she was 6, 5, 5.5, 7. She was all over the place. So with our second one, we didn't really know exactly the conception date. And so we were about, a week or so off. And at that point during a typical pregnancy, well, at least what we thought, the protein markers indicated, some potential genetic conditions associated with trisomy 21 and whatnot. So understanding your cycle would allow you to kind of get a better understanding of that. And, we had to go through genetic screening and whatnot and answer a bunch of weird questions. So no problem. That's why it's important to kind of understand your cycle. Here's what it looks like. Here's a karyotype of somebody with Down syndrome. Okay. And that's what it looks like. There's your 23rd chromosome. 23rd chromosome is what determines sex. So, and there's the X, and it's not really a Y. It's just a small little nub. But that's all you need to become a man, I guess. I don't know. Interesting. You can get changes in any of these, and there's various different conditions and disorders associated with them. You can get a change in just this one right here, and it can result in too many X's or too many Y's. There are a number of them. I just find some of these fascinating. But the one thing that you need to remember, and I think I mentioned this way back when we started this, okay? All you need is this, okay? All you need is a Y. As soon as you see a Y, you know it's a guy. If there's nothing here, and that's what this is saying, there's nothing there. Okay, they're missing half of the genetic material associated with that sex chromosome. Okay, so it's still a female. Okay, because there's no Y. It could be triple X. It could be quadruple X, and those do exist. They have a specific set of kind of hallmark characteristics associated with them. Some of them can, triple X female, an XXX female, she can produce viable eggs, I believe. XXXX, I don't believe can produce. viable offspring. Males can have an extra Y. They can be XYY. They can't be not. They can't be this. They can't be this. Okay. They can't be that. You got to have at least an X. Okay. Because the Y chromosome doesn't have a whole heck of a lot of information on it. I think I have a link maybe in the next lecture that looks at some of those. But the Y chromosome only has really one major thing on it. I think that's listed on the next slide. There's a few things, but really the most important thing is the testosterone, producer. So Turner syndrome is XO. Okay, it is a female. It can happen from non-disjunction. So there's the most common non-disjunction associated with a female sex chromosome number. For a male, it's called Klinefelter syndrome. Okay. And you can see there's an extra X. Okay. You can kind of see, they can produce some really crazy number or really crazy chromosomal situations there. So you wouldn't know. You wouldn't know. I think there was a paper written. There was a newspaper article written, I think, maybe a handful of years ago, following some of these people that had Turners and Klinefelters and whatnot, and some of the other ones. And you wouldn't know that necessarily they had an issue. So because they look pretty normal, and there's so much great therapy now with some of this, with genetic treatments and whatnot, that manipulations with hormone injections and whatnot, that you can still get some of the secondary characteristics associated with a male and a female with it. You can obviously see here underdeveloped testes and prostate. Some breast development can occur with these males, but and this as well, guys, just because there's an extra Y chromosome, because sometimes there's an XYY, that does not mean you have even more testosterone. There's a study that looked at when it was first discovered that like the vast majority, like 90-something% of this prison in Russia, because they did a bunch of genetic research in these prisons over in Russia. And like 90% of everybody in this jail was like XYY. So it was postulated that the extra Y chromosome gives you more testosterone, more rage. But that was just an outlier. They tried to, reproduce that type of study, and it was never shown to do that ever at any other location at all. It just happened to be a thing. But the big thing on that Y chromosome is this right there. Okay. That SRY, that is the testes determining factor. Okay. If that's there, you get a guy. If it's not there, you don't get production of male parts. Okay. And that goes outside and inside the body, as well as outside development as well. And again, you can see there's a karyotype showing that off there. There's a bunch of things that can also happen. There are a number of different changes here. There are 4 different changes that can happen in chromosomal numbers there, where something is either broken off, duplicated, removed, flipped. These make a good matching. They make a good multiple choice question, hint for an exam coming up for this thing. So here are 2 of them here on this slide, and here are 2 of them over here. Deletion, again, if you know the English language, you can probably figure out what in the world these mean. So deletion just means it's removed. It's deleted, okay? It's broken off, okay? It's broken off for whatever reason, okay? Could have been exposure to a teratogen, something that can mess up genes. Could have been exposure to a virus. There's a lot of different causes for this, okay? Duplication, I mean, it's duplicated, it's copied. So you get an extra chromosomal segment. It can be, so there's, see, deleted, boom, deleted. Duplicated, it got duplicated. You can see it could be inversed, okay? Where it's flip-flopped. Translocated. Okay, you can see they've just been flip-flopped. Okay, some of these don't do a damn thing. They don't. These mutations can potentially not code for anything. They can potentially code for stuff, though. Because remember, what is listed in here? This is the information, right? That tells the cell how to make a protein. Okay. When it comes to some of these, like this one and this one, it's not the same. Okay. You're missing stuff. You've added stuff. That makes a total different list of instructions in which to make a protein. It may become totally dysfunctional now. And you see that in sickle cell anemia. Deletion of just one little amino acid takes that round, circular, red blood cell and makes it into a sickle-shaped structure. Here with a translocation or an inversion, the chromosomes are still the exact same length, so there's really no additional or subtraction occurring. But what do I have now? I have a different sequence of amino acids. I have different instructions. And again, it can code for something that is completely off base and totally changes the function. It could code for something else and it doesn't change the function of the protein, okay? They're not all potentially bad. That's what I'm trying to say. Because, this is, this could almost be just crossing over, right? So this is what gives us just a little bit of variation. Doesn't necessarily have to be a bad thing. Although we kind of see it as that. Here's a couple pictures of some of these guys. There are a bunch of them. There's this one, Williams syndrome. And I put this in here because these are some of the more common ones. With Williams syndrome, you've lost the end of chromosome 7. That changes everything for what that region of this chromosome coded for. And it codes for a specific type of protein fiber called elastin. And with these individuals, you get this very characteristic pixie-like look, okay? And that's because of the elastin that is changed within their skin. Another one that we see here is this Argyle syndrome, and this is a translocation. This is one that gives rise to a number of genetic defects, okay? This changes a lot of the connective tissue structures within the body. Connective tissue is literally everywhere, okay? It helps to hold all of this together. Some of the major things that it causes heart issues. And it's called tetralogy of flow. What does that mean? You have two sides of your heart. The left side pumps blood throughout the entire body, okay? It pumps blood up to your head, out to your fingers, down to your toes, all at the same time. It's huge compared to the right side. That is a little bit smaller in size compared to the left side, left chamber. So it doesn't have to push blood with as much force because it's just pushing it out to the lungs that are right here. But with these individuals who've had a translocation between chromosomes 2 and 20, now the right side gets overdeveloped and the left side gets underdeveloped, those two chambers down there. The pulmonary trunk is flip-flopped with the aorta. Again, this is all because of just this two little chromosomes flip-flopping material. Okay. Sometimes they code for stuff and it does not a damn thing. Sometimes it causes some severe issues here. And I know that that's only two things, there's two other things, but you get the idea. The heart just is not up to snuff. So you have to have some major surgeries to repair this sucker. There's some other ones there with chromosomes causing various different types of cancer. One of the more common ones associated with translocation anyway is between 22 and 9. And that's cryogenic myelogenous leukemia. It's just a blood cancer. Okay. I'm going to highlight some things here that you should probably remember. And again, these are just some examples of that. Okay. There's also one called cry du chat, which means cry at the cat. And that's a genetic condition where you have malformations that are occurring within the larynx. And so when these kids cry, that and some tissue flaps